So let's have a look at some of the most recent examples where that has happened. And this is the first example I want to share with you. This is work on the sialylation on the C1 inhibitor glycoprotein, which is a really complex molecule and which has 6N and 7O glycosylation sites. So there are plasma-derived drugs on the market called Berinert and Cinryze. And if one wants to come up with a recombinant version, then the main issue here is half-life. So the company Civic used a genetic engineering approach to help, and they overexpressed different sialyl transferases in their human CAP cell line. And you can see here that the wild type has some sialylation on the O glycans, and if you genetically engineer the cell line, that has already been done on the N glycans, but here it was also done on the O glycans. You can see that you have quite some additional sialylation here on these O glycans. And if you then test for half-life, then you see a very significant increase of half-life compared to the wild type, which you can find here, right? And here is the engineered version, and that's pretty similar to the plasma-derived C1 inhibitor. So you have a huge effect by increasing the sialylation on the O glycans. Still, if we compare these pharmacokinetic data with Fc fusion proteins, this is really still rather short half-lives. So if we have a look, for example, at this Fc fusion protein, that's data from Etanercept, which is in clinical development at the moment against glioblastomas. It's a fusion protein containing the extracellular part of the CD95 and coupled to an Fc domain. And you can see here on the left-hand side how strongly the degree of sialylation influences the clearance by the asialoglycoprotein receptor. So the higher the degree of sialylation, the lower this clearance rate becomes. And you can fit these data and calculate this kind of equation here. And you can use this correlation to build a model to predict the serum concentrations of different batches of this Fc protein. And this here shows you the training data set that was used to generate the model. And here you see the output of the model when you see here the predicted serum concentration and here the observed. So that's a nice tool to not only predict the serum concentrations later on of the different batches of this product, but also derive dose recommendations for clinical trials. In this case, they reported dose recommendations for children. So that's one Fc fusion protein where we've seen a very strong effect of sialylation. And in the next study, from this year actually, this group, Hegel et al. Looked at the importance of the different domains of our Fc fusions for the pharmacokinetics. So what they did is they looked at two Fc fusion proteins, each equipped with one N-glycan in the Fc domain of the protein and two in the fusion part. And as you can see here from these plots here, there were mainly neutral glycans on the Fc part, so not a lot of peaks here, so rather easy to analyze. And then on the fusion part of the molecule, there were a lot of complex and highly sialylated glycans present. And what they did afterwards is that they administered these different variants or of these fusion proteins in a hamster model and looked at the specific clearance over time. So what they did in detail is they were looking for the fusion protein number 1. They tested the Fc part and the fusion part, which they call the receptor part, individually. And for the fusion protein 2, they looked at the whole molecule. So what they found is that the fully galactosylated glycans here, right, decreased the area under the curve by 15 and 19%, and they could also show that the best area under the curve was obtained, but with the monosialylated fusion protein. So that was the glycan variant that was having the best pharmacokinetic behavior. When you look at the different parts of this fusion protein 1, the receptor part and the Fc part, you see that if you only look at the Fc part, none of those variants seem to be cleared faster or slower than the other, which is not the case here for the receptor part. So here it's clear that the A2F variant is the one which is really cleared a lot faster. So it seems that the receptor part seems to be more important than the Fc part when it comes to pharmacokinetics. Yeah, so that's what I wanted to show you about Fc fusions, and now I want to move on to sialylation of IgG, which is something that people haven't looked much into, and so I found this quite interesting. This paper also from this year, it was aiming at the production of highly sialylated Fc glycans in IgGs. Through genetic engineering, and what these people did was introduce different changes in the Fc part. So, for example, a deletion at position 294, which caused loosening of the Fc structure and led to a high degree of sialylation on the Fc. That's what you can see here. So this is the wild type, and this is the engineered version with this deletion, and you see that that there are quite a lot of sialylated glycans appearing here. So that's the desired effect that was achieved here with this deletion. And what they did on top was introduce further mutations in the Fc to increase the affinity to the neonatal receptor. And when they then went into a mouse model with these different IgG variants, the first thing that they observed is that the mutation that was targeted at the receptor enhanced the half-life of the mutated products. So that's what you can see here. This is the wild type, and here are the variants, so slightly increased. And if then on top of this mutation you added the deletion, which produced the higher sialylated product, then you could even have a much higher extension of the half-life of this product. So that's a very nice results here, and that could also show that if you remove the sialic acids from these products, you kind of fall back to the initial level. So this here is the product from with the deletion from the cell line with a deletion. This is the sialylase A treated sample, and this is the product from the wild type. So if we kind of summarize these slides and the effects that the different types of glycans, especially sialylation, have, we can come up with the following conclusions. So if we're looking at complex glycoproteins, the main parameter that one wants to look at is the ratio of sialic acid versus terminal galactose, and to a much lesser extent what happens with regards to high-mannose. So the main focus should be on this here. For the FC proteins, obviously we're adding the affinity towards the neonatal receptor, which is very important. And then again, the ratio of the sialic acid and the terminal galactose is important. Again, high-mannose may have an impact, but it's considered not to be that important. Important to point out here is that it seems that the glycosylation on the fusion part of the molecule seems to be a lot more important than on the FC part. So that is something that was shown on one of the slides before. So when it comes to IgG, I think the main thing to look at is the FcRn affinity again, then the high-mannose. I think that's what's everybody's concern in IgG. And the sialic acid has not, at least to my knowledge, not been considered very important. Maybe that's going to change. So we'll see in the next years how many people are going to look at sialylation on IgGs. So